Hello, my name is Jorge Lizondo, and I will be presenting on Chapter 23, which covers vector nutrition and energy metabolism. So to start off, there are three groups of blood-feeding insects that can be distinguished based on their blood-feeding habits. The first group are species where only the larvae feed on blood, and this is actually very rare. An example of species of this are maggots. For the second group, we have species where they only exclusively feed on blood throughout their life cycle, and this includes species such as tsetse flies, triatomines, and lice. And for the third group, we have species where only the adults feed on blood, and this includes species such as mosquitoes, black flies, sandflies, and fleas. So there is also a difference in which sex feeds on blood. And so this can also be grouped into three categories, the first of which are species where both sexes are obligatory blood feeders, and this includes species such as fleas. The second group involves species where both sexes are optional blood feeders, and this includes species such as stable flies. And the third group involves species where only the female is an optional blood feeder, and this is regarding species such as mosquitoes. One important thing to note about obligatory blood feeders is that they pretty much always harbor symbionts, and so this suggests that the blood meal is not nutritionally adequate. The majority of work on nutrition and energy metabolism in blood-feeding insects focuses on mosquitoes, and in particular, the majority of this work focuses on Aedes aegypti due to the ease of rearing, desiccation-resistant eggs, cosmopolitan distribution, and the vector competence of the species. And so work on nutrition and energy metabolism in blood-feeding insects has led to these three statements, which are that female mosquito reproductive capacity is affected by their nutrition in both larval and adult stages. Female mosquito reproductive potential is established by the end of the larval stage, and subsequent exploitation of that potential depends on adult stage nutrition. And these statements summarize a lot of the studies that have, again, revolved around mosquitoes and females in particular, and there needs to be an understanding of the biochemical mechanisms that validate these statements in mosquito biology. So, for example, if a female mosquito must attain minimal energy reserves, which could be in the form of lipid or glycogen, before they can mature a batch of eggs, their behavior will be oriented towards obtaining those energy reserves. And the energy reserves can come from sources such as a sugar source or blood. And so depending on the preferred energy source, which can be either sugar or blood, this choice or behavior influences disease transmission because disease transmission only occurs when the preferred energy source is blood. Mosquitoes cannot transmit disease if they only feed on nectar. The size of the energy reserves directly determines the longevity of a mosquito. Mosquitoes with a minimal energy reserve may not live long enough to find a host or live the length of time that is required for a pathogen to develop. And the size of the energy reserve also directly affects the disposition of the blood meal. What this means is that if the energy reserves are minimal, the blood meal components may be deaminated and used to increase energy reserves rather than being used for egg production. One key aspect about this is that more than one blood meal may be required to mature the first batch of eggs. So if multiple blood meals are required, this can lead to an increase in disease transmission because more hosts are bitten to satisfy that number of blood meals. So now we move on to larval nutrition. In laboratory conditions, larvae accumulate reserves in proportion to their body size, and so the larger female larvae accumulate more reserves than the smaller males. And larvae seem to have a critical period for nutrient accumulation in which more than 70% of the total protein and 80% of the total lipid is deposited during this period, which is during the fourth and final instar. And instar refers to the stages between molts, and mosquito larvae molt 4 times. So Aedes aegypti are a species that are notable for the large amount of lipid accumulation compared to other chulosine and anopheline mosquitoes, and they display a significant degree of nutritional plasticity during the fourth instar. This means that they're able to respond to starvation and continue feeding and prolonging development, and the mosquito larvae can pupate with significantly different sizes and nutritional reserves. Mosquito larvae are aquatic, so the nutritional quality of their environment varies widely. So due to this, mosquito larvae fall into two main ecological groups. First, we have Anopheles, which inhabit groundwater. in the form of ponds or marshes, and they exhibit rapid development time to accommodate variations in availability of nutrients. And this rapid development time could be due to the pressure exerted by predators that can consume the larvae if they persist in the larval stage for a prolonged period of time. In the second ecological group, we have Aedes and Culex, which inhabit small containers such as tree holes or man-made containers. And these environments are relatively predator-free and nutrient-poor. So due to being nutrient-poor, the larvae develop slowly and can adjust the larval period length to the availability of nutrients. So the Aedes aegypti species is particularly adept at adjusting its larval development to the availability of nutrients. This is because it can develop as fast as any species in a rich nutrient environment, and they can develop in nutrient-poor environments better than most other species. Now we move on to adult nutrition, and regarding adult nutrition, almost all studies have been conducted on the Aedes aegypti species and Anopheles species. And so the results for Aedes aegypti have shown that their extent of nutrient storage was dependent on relative body mass. So larger females have greater energy reserves, and when fed to repletion, large females ingested more blood and produced more eggs than smaller females. And in small females, as little as 2% of the blood meal protein amino acids were converted to egg proteins, and this is because most blood meal protein amino acids are deaminated. And in large females, at most 20% of the blood protein amino acids were converted to egg proteins. Some alpha keto acids derived from the deamination of the blood meal protein amino acids may be used as the carbon source for egg lipid synthesis. Another portion may be used for energy production, and they may be used to synthesize energy reserves. However, no quantitative measurements have been made regarding these hypotheses. So in optimal conditions, when large females are taking large blood meals, no more than 50% of the blood meal protein amino acids were converted to egg components in Aedes aegypti species. However, in suboptimal conditions, when small females were taking small blood meals, no more than 30% of the blood meal protein amino acids were converted to egg components, and the egg components are proteins and lipids. And when large females take small blood meals, they need to use some of their energy reserves to support egg production. But with large meals, there was apparently no use of the female's energy reserves for egg production. In Anopheles, the extent of nutrient storage was also dependent on relative body mass, and so larger females had greater energy reserves. And the Anopheles showed somewhat similar results to Aedes aegypti. The only difference was that they were less efficient than Aedes aegypti in converting blood protein amino acids into egg components, and so due to this, they behave as if they were basically smaller Aedes aegypti. And in Anopheles, at most 20% of meal protein amino acids were converted into egg components, and about 33% of protein amino acids were converted to maternal energy stores, which was more than occurred in Aedes aegypti. And in this particular conversion regarding maternal energy stores, they were actually more efficient than Aedes aegypti, and it is unknown how the rest of the blood meal protein amino acids are utilized by Aedes aegypti. So continuing with adult nutrition, these studies show that the fate of the amino acids derived from the meal protein is complex because some amino acids from the blood meal are used to synthesize egg proteins, but the majority of the amino acids from the blood meal are deaminated. And the deamination of amino acids is when the amino acid is broken down by removing the amino group. And so this releases ammonia as a byproduct, which we'll be covering shortly. But when the amino acids from the blood meal are deaminated, which is the majority of the time, the resulting alpha keto acids can be used for synthesis of egg lipid, synthesis of maternal energy reserves, and energy production. And so here in figure 23.1, we have a depiction of the possible fates of blood meal protein amino acids. TAG stands for triacylglycerol, which can be found in fat body, midgut, or eggs. And glycogen can be found in the fat body, muscle, midgut, and trehalose is present in the hemolymph. So in mosquitoes, which we've been talking about, Aedes aegypti and Anopheles species, sugar feeding is an important factor in regulating the fate of amino acids in the blood meal. So some of the roles of sugar feeding, we have three listed here. For the first one, we have that it facilitates previtallogenic follicle development. And previtallogenic stage in mosquito development refers to the period before the mosquito's ovaries start actively producing yolk proteins for egg development. So for the second role of sugar feeding, it reduces the size of minimal blood meal that will support egg production. And for the third role of sugar feeding, we have that it increases the number of eggs produced. And one thing to note is that sugar feeding does not, however, increase the efficiency of utilization of protein amino acids in the blood meal for the production of egg components beyond 50%. So there is a limitation. So to conclude with adult nutrition, the fate of protein amino acids in the blood meal is intimately tied to the energy status of the female at the time she takes the meal itself. If the female has energy reserves above a critical minimum value, she will develop eggs. And if the female has energy reserves below a critical minimum value, the meal amino acids will be utilized to increase her energy reserves. And the energy reserves depend on larval nutrition and on sugar feeding by the adult. So now we are going to discuss the metabolic problems that are associated with blood meal metabolism. And so based on findings, mosquitoes could have a difficult time dealing with a large increase in amino acid concentration that occurs following the digestion of blood meal proteins, especially if more than 80% is converted to alpha keto acids. So due to the large amount of deamination that occurs, this produces significant amounts of ammonia, and the ammonia itself is excreted by the mosquito as a combination of ammonia, uric acid, and urea. And the amino acid conversion to alpha keto acids itself takes place in either the midgut or the fat body or both of the mosquito. So here we have figure 23.3, which represents the changes in hemolymph-free amino acid composition 24 hours following a blood meal in Aedes aegypti. And so you can see that the concentration of many amino acids does increase following feeding, but specifically there is a very large increase in proline concentration 24 hours after a blood meal in Aedes aegypti, which you can see in the very last bar. So yeah, that result is pretty significant, how the proline concentration increases by a lot compared to the other amino acids. And so after the proline concentration has increased significantly by the 24-hour mark following feeding, there does end up being a steady return to its pre-blood meal level by 96 hours following feeding. And so this begs the question of what is the importance of the massive production of proline following feeding? So the amino acid proline is produced by glutamate, and glutamate itself is a major product of transamination reaction involving an alpha ketoglutarate, which you can see in the figure 23.4 here. So the fate of proline is to then be converted into protein. So proline is basically an innocuous compound. One key aspect, however, is that proline could potentially serve as a reservoir for ammonia, which again we see in figure 23.4. And this could store the large amount of ammonia in a metabolically safe form until it can be converted to urea or uric acid for secretion. As ammonia gets recovered from proline, alpha ketoglutarate can then be used for energy production or for the synthesis of more proline. So uric acid and urea synthesis in large amounts requires up to 15 to 20% of caloric content of blood meal proteins. Comparatively, however, the interconversion of an alpha ketoglutarate and proline takes place without significant energy cost. So basically, these findings do suggest that a proline cycle operates in a mosquito, and this proline cycle is what allows massive deamination of amino acids for energy production while storing that toxic ammonia that's produced as proline. So figure 23.5 here is basically showing that how the findings do suggest that a proline cycle is what the mosquito utilizes to be able to store that toxic ammonia that's produced as proline. So the blood meal proteins are deaminated into amino acids, the majority of them, and they become keto acids, and that produces that leftover toxic ammonia in very large quantities for the mosquito, but it is able to basically store them in a metabolically safe way as proline. So now we move on to energy and reproduction. So in mosquitoes, Their ovarian follicles, they must reach a certain developmental stage before a blood meal can support egg production. So before that's even possible, egg production. And so small females with low energy reserves, they require additional energy to complete maturation of follicles before a blood meal can actually support egg development. So this is where the two main types of meals come from. And so the energy can be derived from either a sugar meal or a blood meal. So sugar meals are more effective than blood meals in building energy reserves in mosquitoes, and the female mosquitoes will often have a sugar meal before their first blood meal in order to accumulate sufficient energy reserves to support egg development following a blood meal. So in field conditions, mosquitoes usually do have little energy reserves for flight and survival. So sugar feeding must occur during the first few days and blood feeding occurs later. Without a sugar meal, the males only survive about 2 to 3 days and the females only survive about 3 to 5 days, again without that sugar meal. So field mosquitoes, large or small, they do have energy reserves that are far below those seen in lab insects, and the energy reserves of the field mosquitoes are close to minimal reserves seen just before death in starved lab mosquitoes. So that shows you the disparity between the two. And so sugar availability could potentially be limiting to fitness in mosquitoes. So therefore, it is important to note that mosquitoes must maintain a balance between sugar meals and blood meals. This presents a dilemma for the mosquito, which only takes a few but very large protein meals. So they must have appropriate partitioning of feeding on either a sugar meal or a blood meal. And this does have a critical impact on their reproduction. So sugar itself is used to accumulate the necessary reserves to support egg development, which then requires a blood meal. So now we move on to flight metabolism. Insect flight muscle is the most metabolically active tissue that is known. The oxygen consumption of the insect flight muscle increases 50 to 100-fold over resting muscle. And so just to offer some comparison, in vertebrates, the increase is only 7 to 14-fold. So that shows you the disparity between the two. And flight itself can be powered by sugar, lipid, or proline metabolism. At max output, a locust flight muscle hydrolyzes ATP at a rate of 18 micromole per second, and the entire energy-rich phosphate pool, which is ATP and phosphoarginine, of the flight muscle can sustain flight for approximately 1 second. So in order to actually sustain this flight, there must obviously be an immense amount of ATP synthesis occurring during flight, and the flight itself in insects can last several hours. So continuing with flight metabolism, in the insect orders Diptera and Hymenoptera, carbohydrate is the main substrate that is used to fuel flight. Unfed mosquitoes, which can range from about 1 to 2 days old, they do have just enough fuel reserves to fly for several hours on a flight meal at a rate of about 1 kilometer per hour. So at these conditions, ATP hydrolysis rate is adjusted to locust body weight. That is about 12 micromoles per second. So they have glycogen stores of muscle that can also sustain flight for a few minutes. And prolonged flight in these insect orders, Diptera and Hymenoptera, it utilizes the glycogen stores of the fat body, and the sugar is then transported to muscle as trehalose. And mosquitoes, they must oxidize about 2.3 micrograms of glycogen per second to produce the required amount of ATP to actually sustain their flight. And so during flight, the mosquito first actually consumes sugar in the crop if it is present, and then it consumes glycogen. And after the mosquitoes have flown to exhaustion, during which glycogen reserves are depleted, they can then immediately fly after they have another sugar meal. Comparatively, though, they can't fly for several hours after a blood meal. So that just goes to show that a sugar meal is valuable as opposed to a blood meal when referring to flight specifically. And that is just simply due to the time needed to convert blood meal protein amino acids into glucose and trehalose. It is interesting to note that proline is the main substrate that fuels flight in some insects by a pathway that involves partial oxidation of proline in the insect flight muscle. And in Aedes aegypti particularly, the free proline concentration in the thorax drops 40 to 60% following about 30 to 60 minutes of flight. And so this suggests that in addition to glucose, proline also serves as a fuel for flight. And here we have figure 23.6, which shows that proline is also used to fuel flight metabolism. And you can see there towards the bottom how proline is produced in the fat body from glutamate, and the proline itself is transported to the flight muscle where the carbon is converted back to alpha ketoglutarate, and the alpha ketoglutarate then goes through part of the citric acid cycle to form malate. And there is synthesis of ATP, and we see that the partial oxidation of proline in the flight muscle yields 14 ATP exactly. And now we are going to discuss autogeny. And so autogeny refers to egg production in absence of protein intake by the adult female. And so autogeny shifts the responsibility for gathering nutrients for egg production from the adult stage to the larval stage instead. And it is likely that storage proteins in the larva and perhaps proline play important roles in providing the amino acids that are required for egg production. And in some, but not all, autogenous mosquitoes, sugar feeding by the adult is required before ovarian development can actually begin. And anautogeny refers to egg production that is dependent on protein intake, unlike autogeny. And to summarize, the manner in which female mosquitoes digest a blood meal has important implications in all aspects of mosquito biology. And so future research has shown that there is an importance of micronutrients or non-energetic nutrients, or NEN. They are needed in reduced amounts. So they are essential for many mosquito physiological processes that influence biological traits, such as vector competence and reproductive capacity. And some of the non-energetic nutrients are amino acids, vitamins, salts, metals, and sterols. So we discussed amino acids, but in addition, again, vitamins, salts, metals, and sterols. And in particular, it was found that iron, calcium, salts, sterol, and vitamin homeostasis is needed in order to sustain respiration, cell signaling, metabolism, and reproduction in mosquitoes. And micronutrient homeostasis does have implications on mosquito immunity and vector competence overall. Thank you very much, and these are my references.